Hello and welcome. My name is Carmen Ugarte. I'm in the Department of Natural Resources and Environmental Sciences at the University of Illinois. As part of the outreach component of the Corn and Soil Health Project, I will introduce you to a series of virtual field days on a series titled Ears to the Ground. We wanted to start this series by talking about measuring and managing soil nitrogen and exploiting biologically based fertility. There are many different ways to estimate soil nitrogen supply potential, and we can simplify this a bit by dividing tests into two categories. First, we have the measures of nitrogen or organic matter stocks, and you may have heard about measures that quantify the active organic matter fraction, such as PAM and POC, for example. There are also measures that quantify nitrogen that's derived from amino sugars. And in this sense, you will hear about indicators like the ISNT or the SLAN test. Then we have other measures that quantify directly nitrate levels in the soil, like PSNT. On the other hand, we have measures of N-mineralization or biological activity. N-mineralization can be quantified in what's known as potentially mineralizable nitrogen, and there are protocols for aerobic as well as anaerobic incubations. In this category of measures of N-mineralization or biological activity, we also find indicators like the Solvida test that measures soil respiration. Here in Illinois, as part of this project, we are studying some of these tests in an on-farm study. But before we get into some of our results, we wanted to visit with folks who are using some of these measures so that they can explain how you could apply them to your particular system. We will hear again about the use of the ISNT, Solvida, SLAN, and PSNT tests. They will both explain the measures that they conduct in their labs, and we hope that you will learn more about how to interpret nitrogen tests, and also that you would expand your thinking on issues of sampling. Both of our guests suggest that care must be taken when considering the timing as well as the depth of sampling. So, welcome to this series, and after you hear from our two guests, you will see a subsequent video in which we will be discussing some of our results coming out from our own on-farm study.